Greetings everyone! Today, I am going to explain the DNA extraction protocol in plants, focusing on the CTAB method. DNA extraction from plants is more difficult than from animal cells because plant cells have a rigid cellulose cell wall and contain contaminants like polysaccharides, polyphenol, tannins, proteins, and lipids, which interfere with DNA isolation. Therefore, fresh leaves that are about 15 to 20 days old are usually preferred, and the tissue is ground using a mortar and pestle or liquid nitrogen. The basic principle of DNA extraction is to break the cell wall, cell membrane, and nuclear membrane, release the DNA into solution, remove contaminants, and finally precipitate purified DNA. The most common method used is the CTAB method. The CTAB extraction buffer contains CTAB, NaCl, tris, EDTA, PVP, and beta-mercaptoethanol, and each component has a specific role. CTAB is a cationic detergent that dissolves cell and nuclear membranes by forming micelles. It also removes polysaccharides and denatures proteins. At high salt concentration, CTAB binds polysaccharides, allowing them to be separated from DNA during chloroform extraction. Sodium chloride, or NaCl, provides the ionic strength needed for CTAB to work effectively. It helps remove proteins bound to DNA, prevents proteins from precipitating with DNA, and aids in eliminating polysaccharides. Tris acts as a buffer, maintaining the pH around 8. This protects DNA from degradation and keeps it stable throughout the extraction process. EDTA chelates magnesium and calcium ions. Since DNase enzymes require magnesium ions to function, EDTA inhibits these enzymes and protects DNA from being degraded. Beta-mercaptoethanol is a strong reducing agent. It breaks disulfide bonds in proteins, denatures them, and also removes tannins and polyphenol, improving the quality of the extracted DNA. PVP, or polyvinylpyrrolidone, binds polyphenol through hydrogen bonding. This prevents them from oxidizing into quinones, which could otherwise bind to DNA and interfere with downstream applications. Finally, phenol, chloroform, and isoamyl alcohol are added. Phenol denatures proteins, chloroform improves phase separation and removes contaminants, while isoamyl alcohol reduces foaming. After centrifugation, the purified DNA remains in the aqueous phase, and is then precipitated using alcohol. In conclusion, the CTAB method is a highly effective technique for extracting high-quality plant DNA, because each reagent plays a specific role in removing contaminants while protecting the DNA.